comments or questions, just like yell it out.
We'll be
All right.
monitoring.
Okay. All right. So I don't think the majority of people on here know me. So I'll introduce myself briefly. I'm Shanice, and I'm a graduate student in the micro department in Georgia Tools Lab. And I'm really excited today to present this paper looking at plant microbe interactions. And Really, this journal club, I'll say up front, is a little bit different will be a little bit different from past journal clubs because it has a little bit more focus on microbiology, but hopefully people found the paper interesting and we're ready to discuss the findings. All right, so in today's paper, the authors looked at how plants are able to regulate the beneficial microbes in the rhizosphere, and specifically how this regulation takes place through the production of reactive oxygen species and how this is mediated through the FAIR system. So here's just a kind of a model that the authors present at the end of the paper, where FAIR is able to regulate the production of reactive oxygen species, and that in turn inhibits Pseudomonas enrichment. And we'll talk more about the mechanism by which the authors present that this might be happening. All right, so a little bit of introduction. So we can all appreciate that plants are associated with communities of microbes, and these microbial communities can be found on the above ground parts of the plant, so the phyllosphere, as well as they can be found intimately associated with the roots in the rhizosphere. And it turns out that there's quite a bit of diversity of microbes that are found both in the phyllosphere and the rhizosphere. So here is just a pie chart taken from this review looking at some of the phylum of bacteria present in the phyllosphere and rhizosphere. And what you can appreciate from just first glance is that there's quite a bit of overlap in terms of the bacteria that are present between the phyllosphere and the rhizosphere, right? So there's a large similarity, but you can also see that there are differences. So, for example, in the rhizosphere, you have things like nitrospirae, which is colored here in dark red, and it is not present in the phyllosphere. And then the same is true for the phyllosphere, where you have chlamydia that is not present in the rhizosphere. Right, so there's quite a bit of overlap, but of course, there's some distinction between the microbes present. And in the last maybe 5 to 10 years or so, I'll say that the plant microbiome has really been appreciated for its ability to confer a fitness advantage. And this is particularly important when we think about climate change and the increasing world population and how we need to harness the microbiomes of plants in order to increase world food production as a result of the population increasing. All right, so I talked about microbes or the microbial community conferring a fitness advantage, and one fitness advantage that it confers is that it affects plant growth. So here from this paper is a plant radish, a 17-day-old plant radish that was colonized with Pseudomonas corrugata, or not colonized with Pseudomonas corrugata. And what you can see very distinctly is the development of the plant root when it is colonized with the bacterium as compared to when it doesn't have the bacterium present. So there's some beneficial relationship as a result of having this bacterial species present. In addition to microbes affecting plant growth, it is also known that microbes protect plants from abiotic stress. And one abiotic stress that that microbes protect plants from is from the stress of drought, which is particularly important in climate change. So, in this paper that was in a study that was done by Devin Coleman Derry's group, they looked at the enrichment of this bacterium Streptomyces in the rhizosphere of sorghum plants. So here is a sorghum plant root that is fluorescently labeled with M. cherry. And this is the control. And then here is just the bright field and the fluorescent image overlaid. And then in drought conditions, what you can see is that there's an increase in fluorescence signal as compared to our control. And then when that is quantified, here shown as fluorescence density, we see that there's an increase in streptomyces as compared to our control conditions. And this makes sense because streptomyces being a gram positive, it is able to basically tolerate desiccation as a result of having a thick cell wall. So you see that enrichment present in drought conditions. And then perhaps most importantly, streptomyces is able to control the growth of pathogenic bacteria, right? So it's able to outcompete pathogenic bacteria, so plants are not susceptible to them, and particularly when they're in these stressful environments. All right, so another bacterium that is enriched in the rhizosphere of plants is Pseudomonas fluorescens. And here again is a Arabidopsis plant root that is colonized with Pseudomonas fluorescens, which is GFP labeled. And just a little bit about Pseudomonas fluorescens for people who are not familiar. And I'm going into this because it is the main focus of the paper. It is a gram negative. It is widely found in agricultural settings. And as the introduction pointed out, it is enriched in disease suppressive plants and it promotes plant health. And here's just an image of what Pseudomonas fluorescens look like. It's a rod-shaped bacterium, and it has these appendages extending from the cell body known as the flagellum that confers motility. So, with this appreciation that the plants are intimately associated with microbes, the authors wanted to ask the very central question: what are the genetic regulators and mechanisms used by plants to control the abundance of beneficial microbes? And it turns out that the immune system is important in this. There's an interplay between the immune system and the plant microbiota. Oops. So as we know, the immune system is able to recognize microbial pathogens and terminate growth. It is also able to engage in plant microbe interactions. And then interestingly, it is able to influence host colonization by beneficial microbes. And I'll say that somebody who doesn't think too much about plants and certainly doesn't think about plant immunity a whole lot, this was really interesting to me. I've never really thought about the immune system as, controlling beneficial microbes. I really, mostly thought about it as, if there's a pathogen, wipe it out, more or less.
Sorry, may I have a question? Quick question. So,
Sure.
These bacteria, they are outside of the plant root, right? I mean, they are not somehow inside. They are always on the surface, right?
So what I'll say, I don't know if always. So in the case in I'm talking about here, they're in the rhizosphere. So yes, outside, but there are definitely examples of like bacteria being present in the endosphere, so inside plant leaves and so on.
Okay.
All right. Yeah, so just to kind of set the stage of for the paper, this is kind of just a 30,000 overview foot view of the plant immune system. So as we know, plants, they are, somebody mentioned that they don't run away. And, that in of itself is a disadvantage sometimes because it doesn't run away. It needs to defend itself from pathogens. And one way it's able to do this is that it has a very robust immune system. So these cell receptors present on plants are able to recognize these microbial associated molecular patterns. In this one example is that it can recognize a flagellum, for example, and then mount an immune response. It turns out that bacteria, for example, can actually secrete some of these effectors through the type 3 secretion system into the plant cell, and then those effectors will in turn bind to these NLRs, and then that can trigger an intracellular immune response. And one cell surface receptor that the paper delves into and is the kind of the focus here today is FAIR. And FAIR is a receptor kinase that's an important part of the immune system. And FAIR, it has malactin-like domain. Most of the reviews and studies that I found said that it has two domains, but for some reason the paper today only depicted it as having one domain. And then importantly, it has this kinase domain. And what I'll tell you is that FAIR seems to be involved in a lot of things. And those lots of things really culminate into it being important in plant development and being important in stress response. And then FAIR is able to bind to the cysteine rich peptides known as RALFs. I think it stands for rapid alkalinization factors. And then, FAIR can then in turn, promote reactive oxygen species, resulting in high levels of RAS, and then that in turn lowers bacterial abundance. But however, if you have RALF present, that can actually negatively regulate FAIR. So I'll talk about that towards the end of the paper. All right, so kind of diving in. So the first question that the authors wanted to ask is, what are the plant genes that regulate beneficial Pseudomonas species in the rhizosphere? And the approach that they took to answering this question was that they rescreened the Arabidopsis mutant library, 16 HSM mutants, and these mutants affected immunity and they affected hormone crosstalk. And specifically, what they were looking for in their screen was their ability of these mutants to support growth of Pseudomonas fluorescens. And the approach that they took I would say, a semi-high throughput approach where they use this nodobiotic plant rhizosphere microbe interaction system, where they had a 48-well dish, and in each well they had these Teflon mesh on which they placed the seeds, and the seeds was really just to separate the shoots from the roots. And then in the bottom of the well, they had MS media with 2% sucrose. So this was 0 days after germination. After 10 days, what they did was to change the media and then add more media, this time without sucrose. And then 2 days later, they inoculated with bacteria. And then after that, they measured luciferase signals. I should have prefaced this by saying that the Pseudomonas fluorescens has a lux operon, so they're able to measure luciferase signal and use that as a readout for CFUs, so colony forming units. So the first thing that they did was that they generated a standard curve to determine CFUs from photo counts. So here they added known amounts of bacteria. So this is CFU. They added known amounts of bacteria and then measured the relative luciferase photo count. So the idea is that when they set up their nodobiotic system and then when they get their luciferase count, they're able to extrapolate and then get the CFUs. Okay, so just as a test for their system and to make sure that the lux operon was not affecting root density, they looked at that. So here's wild type with buffer, and then here's wild type with WCS365. So this is just the bacterium without the lux operon, and then the bacteria with the lux operon, and they saw that there was basically no difference in lateral root density with the lux operon. And then here's just a quantification of that. So this is lateral root density for buffer. We see that there's less, but with the lux operon present, there's no difference. So this was kind of just a check. Okay, so they did their screen, and here again, I'm showing you relative amount of Pseudomonas fluorescens in the rhizosphere, and this again was done in their nodobiotic system with wild type. And they looked at the 16 different HSM mutants, and they saw this, they got this hit that I think they mentioned in the paper that was consistently high. So the levels of Pseudomonas in this, with this mutant was consistently higher as compared to wild type. So this is HSM 13, and this is kind of, the focus for the rest of the paper. So the first thing that they did was that they wanted to see if they, they got this result in a nodobiotic system, meaning that there are no other microbes present. So they wanted to see if in a complex environment where you have other microbes, other bacteria, such as in natural soil, if you would actually still get an enrichment of this bacterium, of Pseudomonas fluorescens. So what they did was that they did the experiment, but this time they did it in natural soil, and then they plated on Kingsby medium. So they're looking for Pseudomonas fluorescens. In the bowl soil, we saw not very much Pseudomonas fluorescens. For the wild type, we see some. But then for the HSM13, we see this significant increase. I think they mentioned that it was enriched more than tenfold in the mutant as compared to wild type, right? And really, this is mirroring what they saw in their screen in their nodobiotic system, that you're able to get an enrichment in a complex soil environment. And here's just a quantification of that. So this is log CFU. For bulk soil, we don't see much. For wild type, we see some. And then for the HSM13, we see an increase in the amount of bacteria or in the amount of Pseudomonas fluorescens as compared to wild type. Okay, so they looked at the morphology for this HSM13 and they saw that it had stunted growth. So here's wild type, and HSM13, in addition to having stunted root growth, also had a hair elongation defect. And these other mutants, this BRE15 and this ARC1, also had defects in, these are mutants that have known defects in elongation. So for the BRE15, they mentioned in the paper that this is unable to perceive the growth hormone, brasinolide, and this ARC1 is a root hair mutant. I think it has defects in microtubule dynamics. So the reason why they tested these mutants is that they wanted to make sure that the enrichment that they were seeing in their HSM13 was not because they have this weird morphology, right? So that's why they looked at these other mutants, this ARC1 and this BRE15. And what they saw for for the known mutants, the BRE15 and the ARC1, was that there was not as much bacterium or not as much Pseudomonas fluorescens as compared to the HSM13, right? So really the enrichment that they're seeing is not because they've somehow screwed up the morphology of the roots. Okay, and then here again is just a quantification. Again, log CFU for the HSM13, there's an increase. And then for the mutants, we see that it's not significantly different from wild type. All right, so they next wanted to map where the HSM13 mutant was, and they did some sequence analysis. So they crossed the HSM13 with wild type, and they got some SNPs. So they got some SNPs in the kinase domain. And one of the mutation was this glycine 598 to a glutamate, right? So you have this small amino acid uncharged going to something that is bulky, relatively bulky and charged, right? So suggesting that there's some disruption of perhaps kinase activity and in turn disruption of this FAIR protein. And that potentially is what is causing the enrichment of Pseudomonas fluorescens. Okay, so I should mention that FAIR4 is another mutant that was previously identified that was present in the malectin domain. And when they looked at morphology, they saw that it had stunted growth similar to the HSM13. And then they wanted to determine if this FAIR4 mutant was allelic to the HSM13. So they did a cross with FAIR4 and HSM13 and saw that they still got stunted growth. And then they also saw similar levels of this bacterium with just the HSM13. And from that data, they concluded that HSM13, FAIR4 is allelic to HSM13. All right. So, oops, sorry. So just kind of a proof of principle, they overexpressed FAIR, GFP labeled, into the FAIR4 mutant, and then saw that there was a decrease in the log CFU of Pseudomonas fluorescens, right? So we know that there's an increase in HSM13 when the bacteria increase in this mutant, and there's also an increase in FAIR4, but when they put back a functional copy, they saw that that increase goes away. And then in FAIR5, which was previously, which is downstream of the kinase domain, they saw this partial increase in the bacteria present. So if we compare the wild type with FAIR5, we see that the levels are not as high as it is for HSM13 or for FAIR4. And this makes sense in light of previous work that showed that FAIR5 is a partial loss of function allele. All right, so from those data so far, they conclude that HSM13, hereafter referred to as FAIR8, carries a loss of function mutation in FAIR, resulting in stunting and increase in Pseudomonas growth. So for the rest of the paper, I'll refer to this as FAIR8. And I don't know if there's some kind of way to how they're numbering or naming these, like why is it FAIR8 as opposed to like FAIR7? Maybe it's like taken, I don't know. This is like,
I think it's just the order they isolated the alleles in. They just number them sequentially based on when they're isolated.
Okay.
That's not always followed in Arabidopsis, though. No.
Yeah. I mean, and often if you have multiple family members, it'd be so FAIR, if you only have one gene named FAIR, then it doesn't have like immediate ones, which is why you get -8. And so the dash indicates it's a different allele coming afterwards.
I see. Okay.
But I'd say more commonly, you might have seen something that say like FAIR 1-8. And then you might have a FAIR 2 would be a different FAIR family gene or from the same mutant screen or something.
Yeah, the problem, of course, is that FER stands for ferritin genes, 1, 3, 4. Yeah. But when they named Feronia, they didn't pay any attention to that.
Right. And there is only one Feronia, right? But there's many Feronia family members. Is that correct?
Yes.
Okay.
But yeah, the rule historically was you weren't supposed to use the same 3-letter code. Whoever had the first use of it had precedence, but I don't think the websites always kept up that well over time. So you had to actually do a bunch of personal searching to find out if there was a same 3-letter code.
Okay. All right. Well, I guess that keeps it interesting. All right. Right. So that's their conclusion from figure 1, right? That this FAIR is a loss of function and this is increasing Pseudomonas growth and causing stunting. So the next question that they ask is, what is the effect of FAIR8 mutation on the rhizosphere microbiome composition, right? So could this mutation be affecting other, just like the makeup of the microbiome? And to answer this question, what they did was to grew FAIR8 as well as wild type plants in the presence of natural soil microbiota. And what they found when they did this analysis was that there was a distinction between the microbiomes of wild type and FAIR8. So here we see that FAIR8 and wild type, they cluster, very distinctly. So the microbiomes are very distinct for these plants. They also saw that there was no shift at the phylum level in FAIR8 as compared to wild type. So this is relative abundance. And here I should just preface this by saying they did 16S rRNA profiling, and they're looking at relative abundance, and here are the different taxa. And if you look at wild type versus FAIR8, there's really, no difference between the phylum, right? So, and I think they mentioned in the paper that, there's no dysbiosis that is seen. So this mutation is not causing any drastic shifts in the microbial composition. However, when they looked at richness and diversity, they saw a distinction. So this is number of OTUs. And for the FAIR8 mutant, they saw that the number of operational taxonomic units was less as compared to wild type. And then when they looked at diversity, so who is there and how much of who is there, they saw that FER8 had less diversity as compared to wild type. And what I will say is that I think I took out these data, but they saw that there was an enrichment of Pseudomonas present in FER8 as compared to wild type. And this kind of led them to ask, is FER8 enriching for beneficial Pseudomonads, or is it enriching for just all Pseudomonads in general? And to answer this question, again, they turned to their nodobiotic system and they looked at commensals. So these are the 5 different commensals, so different Pseudomonas species. And they looked at it with wild type and in their FER8 mutant. And what they saw by and large was that there was an increase in the amount of Pseudomonas with FER8, right, across the board, with the exception of the CH267. When they looked at some Pseudomonas pathogens, so they looked at 4 different pathogens, PA1, Pseudomonas syringae, they also saw this increase, right, with FER8, right, suggesting that FER8 is not necessarily enriching just for beneficial Pseudomonas, but it seems to be enriching for most Pseudomonas species in general. And then as a control, they also looked at non-Pseudomonas species. So here they looked at Burkholderia, 2 different species of Burkholderia, and they also looked at Bacillus subtilis. And interestingly, for Burkholderia, so these non-Pseudomonas species, in some cases, they also saw an increase in the levels of these non-Pseudomonas species. Although I think they mentioned in the paper that the levels were either twofold or less, so not as much increase as it was for the Pseudomonas species. And then certainly for the Bacillus subtilis, you see here in this case a decrease. Right, so from these data, they're concluding that by and large, this FER8 is, enriching for most Pseudomonas species. So then the next question becomes, is the FER8 microbiome beneficial? And I really liked this experiment that they did, where they basically performed microbiome transplant experiments. So here, what they did, they had these first generation parental line and a first generation of FER8, right? And we know that there's a decrease in the morphology of the FER8 mutants are, they don't look as healthy as the wild type. And then we also know that in a FER8, when we quantify Pseudomonas present, we see an increase in Pseudomonas. So this was, they knew this, that there's an increase in Pseudomonas present. But what they then did, what I thought was kind of cool, was that they took the soil from these first generation the parental line, as well as from the FER8, and then they planted wild type Arabidopsis plants. And then they looked at first, the fresh weight of these G2 plants. And you can see, very strikingly that there's an increase in the weight of these second generation plants as compared to the parental plant, really suggesting that the microbiome of FER8 is promoting plant growth. And I guess, one thing I would have liked to see that they didn't put here is that I would have wanted to see if there was a further increase in enrichment of Pseudomonas in the second generation of plants, but that's something that they didn't show. But what they did—
I'll interrupt briefly.
Yes.
Maybe one of the interesting things, if I remember correctly, is that plants for beneficials can become non-beneficial if they get above a certain concentration. So the immune system of plants is important for sometimes maintaining the level of the microbes in the rhizosphere associated with plants to a level where they're still beneficial but not harmful.
I see. So yeah, important in just like maintaining homeostasis, more or less.
So I think that's partially, even here, it's sort of, they were trying to potentially get at that a little bit.
Yeah.
Because again, if you do have this really high level, is that potentially now becoming non-beneficial for the plant?
Yeah. So I guess, and that's a great point. And I guess I would have liked them to quantify here in this, second generation, what the CFUs were like to
Mhm.
see if it was just constant or if there was any,
Yeah.
change.
Yeah, that was partially what's making me, your comment about that was making me think about this idea that things can become non-beneficial at a certain level.
Yeah.
So the other question I have is, so the FAIR8 mutant is quite a bit smaller than wild type. So my question is, are the microbes helping it be as large as it is? So if you were to grow it in absolutely sterile soil, is it actually worse off?
If you grow the FAIR8 in sterile soil, that would be my prediction based on these data.
But they haven't shown that.
I haven't, no.
Okay.
Well, there's one other complication there. I don't know what proportion of the nutrients plant growth was going to pull out of the soil. But the fair plants are littler, so presumably there would be more nutrients left, not just different microbiota, but the nutrient pool available for the Columbia growth in the second generation. There would be less soil depletion with the smaller Feronia plants than with the larger Columbia plants.
I see. So there's a correlation between plant size and their nutrient uptake?
Well, I think a bigger plant is going to have more nutrients in it than a littler plant.
Yeah.
So I don't know if that would have entered into this.
Maybe they could have, I don't know, saturated with all the micronutrients that are needed to avoid that, right?
Yeah, fertilize them essentially.
Yeah.
I don't know if it said that in the methods, but that would be a way around it.
Yeah. Well, I suspect if they watered them with MS or something like that, there's no shortage of nutrients.
Yeah, that's a good point. I'm not sure if when they did these studies in the natural soils, if they watered them with MS. I know in the notobiotic system they did, but. All right, cool. So the next thing that they looked at was whether or not there was a correlation between Pseudomonas abundans in these first generation plants and the biomass of the second generation plants. And they did in fact see a positive correlation. So here is shoot a fresh weight of G2 plants as a function of fluorescent Pseudomonas of G1 plants. And you see that the slope of the line is in the positive direction, right? So if you increase your fluorescent Pseudomonas in the G1 plants, there's a likelihood that your fresh weight will also go up. So the next question that they had was that they wanted to get at mechanism and get at, if there are any transcriptional changes in the FAIR8 mutant that might explain this pseudomonad enrichment. And to do this, they performed transcriptional analysis of the shoots and roots of these plants. And what they found in their analysis was that there were 675 genes that were upregulated in shoots, and there were 82 that were upregulated in the roots. But what was kind of, I think, disappointing was that there was no enrichment of any pathways for the genes that were upregulated in the roots, which was surprising, thinking that they're looking at, the rhizosphere and again, the microbiome that's associated with the plant roots and not seeing any enrichment of certain pathways. I think they were surprised by that finding. However, they looked at the genes that were upregulated in the shoots and they saw enrichment of some pathways. So here is that data. So they saw an enrichment in defense responses, enrichment in stress, and also enrichment in jasmonic acid signaling. And specifically for an enrichment of the defense response, one way they tested this was that they looked at FER8 and its resistance to the necrotrophic pathogen B. cinerea, where in the presence of B. cinerea in the parental line, you see the presence of these lesions. However, in a FER8 mutant, you see that these lesions are more or less, they're a lot smaller, and in some cases, it seems as if they're non-existent. And this is just a quantification of the lesion diameter. Right? So in this FER8 plant, it is, conferring some kind of benefit and not allowing it to be susceptible to pathogens. So they wanted to ask the follow-up question, is this resistance to B. cinerea due to jasmonic acid signaling? And, even though they didn't see any enrichment of jasmonic acid signaling in roots, they hypothesized that perhaps it's the shoot-to-root jasmonic acid signaling that could affect this rhizosphere microbiome composition. So the idea is that you see enrichment of jasmonic acid defenses in the shoots, and then perhaps that might be affecting root jasmonic acid signaling. So to test that hypothesis, what they did was that they made this COI116 mutant in a FER8 mutant background. So this double deletion, and this COI116 is known to be deficient in jasmonic acid perception. And they looked at shoot fresh weights. So here it is for wild type for the parental line. For the FER8 mutant, we see a decrease in shoot weight as we would expect. And then for the COI116 FER8 double mutant, we see that decrease is not as much as FER8, right? So this decrease that they're seeing, they're attributing it to this COI1 gene being present, right? So, they conclude that this double mutant is suppressing the stunting phenotype in FER8. But what was, I think, disappointing was that when they looked at the CFUs in this double mutant, they actually did not see a significant decrease in the levels of CFUs in the double mutant as compared to FER8. Okay, so then they know that the FER system is able to regulate reactive oxygen species. And as I mentioned in the beginning, it is able to regulate reactive oxygen species through through routes. So these routes act as these negative regulators of FER. And these are routes are the cysteine-rich peptides that when bind to FER is able to inactivate or negatively regulate RAP2, and then RAP2 is not able to act through the NADPH oxidases to then result in reactive oxygen species being produced. So the question then becomes, is the loss of FER is that able to induce a decrease in reactive oxygen species result in Pseudomonas overgrowth, right? So if you're able to inactivate FER, you're able to inactivate FER, you're able to decrease RAS. And then as a result of having low RAS levels in the cell, then you get this Pseudomonas enrichment. So the first thing that they did was they had a dye that they're able to look at RAS presence on the roots of these Arabidopsis plants. So here is buffer in the parental line. You see the production of RAS as indicated by these colors. And in the FER8 mutant, you can appreciate that the levels of RAS are a lot lower as compared to our control. And here is just a quantification of the average signal intensity. So in the FER8 mutant, you have a decrease in RAS levels. And then this other experiment that I'm not sure if anyone has any insight on why they did this, but what they did was to add bacterial cells, so in this case Pseudomonas fluorescens, to both the parental line as well as to FAIR8, and they saw again this added decrease when they added the bacterium. And I might have missed something, but I'm still not sure as to why they did this experiment. Why did they add the bacterium to see if ROS levels were decreased? Is it known in the field that adding bacteria decreases ROS? I kind of thought as ROS affecting bacteria, not kind of the other way.
Yeah, I'm not sure that, and that's a beneficial, that WCS.
Yeah, this is the fluorescence.
Well, it's sort of seeing these data would imply that there's something independent of Feronia that is responding to the bacteria reducing ROS levels in the plant. Right? That's what these data would implicate.
Yeah.
So it's working through a different receptor.
I see.
So one thing in the root, the cell surface receptor, the MAMS and DAMs, they are weakly activated in the root compared to the shoot. So in the root, the most defense system rely on the ROS to inhibit the pathogen growth, and these beneficial microbes, they have a way to suppress the ROS just to colonize in the rhizosphere. Okay. So this is a defense mechanism by the plant in the root, and the beneficial microbe can suppress that. Yeah. Okay.
So do they do that through effector sledding? Do you know?
I don't know. I don't know how they do that. Yeah.
Yeah, I guess it would have been nice maybe if they maybe looked at on the microbe side, if they mutated some genes involved in ROS, and then see if this decrease would go away.
But it may not have anything to do with ROS production in the microbe. It may actually just have to do with the microbe suppressing the ROS production in the plant.
Yeah, I guess that's what I'm saying. If like you could mutate, whatever mechanism by which microbes are able to inhibit ROS in plants.
If they know that, right? Yeah.
Yeah. All right.
But they did look at plants that can't make ROS, and they saw more pseudomonads, right?
Mm-hmm. Yeah. Okay. Right. So this is kind of what Mary Lou is alluding to. So they looked at these mutants that were defective in. They dampen basically ROS production. So this RBOHD and RBOHE mutant. So here again is log CFU for wild type. For the FAIR8 mutants, you see this increase in bacteria. And then for these two mutants, RBOHD and RBOHF, you still got that, we still see that there's an increase.
Would have been nice had they been able to test the RBOHDF double mutant. You'd sort of wonder if that was additive and get us up to the FAIR8 level.
Yeah, agreed.
So Anna, they act in the same pathway? RBOHD and F?
Well, there are two respiratory burst oxidases, and if they got up to the FAIR8 level, it would say that FAIR8 is acting probably exclusively through those two.
Yeah, okay. So they also looked at, so FAIR is known to interact with immune receptors and basically allow for, complex formation of these immune receptors. So they looked at EFR1 as well as FLS2 and BAC1-5. And they saw that there was basically no difference in these mutants as compared to the parental strain, right? So it's not acting by scaffolding some of these other receptors that FAIR is known to act through.
Although just to add on, it's sort of the thing we had last week. It would have been nice to do the double and triple mutants because clearly they're not necessary, but could any of them be sufficient? So you'd have to get rid of them all I think.
But isn't BAC1 the sort of doesn't it do all the sort of heterodimerization with EFR and FLS1? And so if you get rid of BAC1, it's like it's like making a double mutant. I'm vaguely remembering something like that.
Yes, you're right. Yeah. And in the suit, the Feronia, it acts as a co-receptor. It induces the complex formation between BAC1, FLS2, and EFR1. And that is activate the defense response in the suit. So
it's taking them both out.
Yeah, so in theory, by just knocking out BAC1, you should be okay.
Then they made a double with BAC1 and BKK1. That's a supplement.
Yeah, that is a BAC1 like brother of BAC1.
Okay, they don't need them.
Okay. All right, so yeah, it doesn't need them. So I kind of mentioned that FAIR is able to regulate ROS through these cysteine-rich peptides known as RALF. And FAIR is known to positively regulate RAP2 activity and that in turn positively regulates NADPH oxidase. So they wanted to ask then, does FAIR act through these NADPH oxidase to regulate rhizosphere pseudomonads? And what they did was that they overexpressed RAP2 in a FAIR8 mutant and saw a decrease in CFUs when they overexpress RAP2 in that strain. So here again is FAIR8, we see that increase in CFUs. And then when RAP2 is overexpressed in that background, we see that there's a decrease, right? So that's what we're looking at. But then what's interesting is that they mentioned that when they look at ROS production in these mutants, and these are just 3 different lines, they saw that the ROS production was actually increased as compared to the FAIR8 mutant. And I guess that makes sense, right? Because if you're increasing or if you're decreasing your CFUs, that decrease in CFUs because you have more ROS. But it doesn't quite track, right? Because if you look at line 3-1, right, in some ways, this is like very comparable to FAIR8 to me. But at the same time, you see a decrease in CFU for the 3-1, right? So it doesn't quite match up with what you would expect quantitatively. So that's the RAP2 overexpression in the FAIR8 mutants. They also just looked at RAP2 overexpression, and indeed, just like in the FAIR8 background, they saw a decrease in bacteria. So we're comparing this as compared to FAIR8, and we see a decrease. And then when we look at ROS production in RAP2 overexpression, we see that there's an increase, right? So if we compare FAIR8 here, for the 5-1, we see an increase, And for the A2, maybe there's a decrease. It looks pretty similar to me. Yeah, there's an increase. Yes. Yes, there's an increase.
These experiments are hard to to be very quantitative about, because when you overexpress a G protein, it has all these other regulators, right? So that doesn't mean you're making the constitutively active, which is really what you would have wanted to do, is to overexpress the constitutively active version so that it's just the on version. But even that is a dangerous thing and may not be tolerated by the plant. So it's just a little bit hard to be super quantitative with these experiments, I would say.
Okay. It's also interesting that, overexpressing RAP2 in the absence of a FAIR8 mutant seem to have there's more decrease as compared to when it's in the FAIR8 mutant background, right? So if you compare like these bars as opposed to this.
Yeah, I think that the ROP2 overexpression by itself should probably be compared to Columbia by itself, in which case it's not really showing a difference. So it's really only showing a difference. So it's pretty close to a wild type phenotype normally, but it does cause a partial reversion of the FAIR8 phenotype.
So I guess, why would you compare it to the Columbia? This is probably just my own.
Just because Columbia is the basic control for what a normal plant, quote unquote, normal plant would be looking like. So Columbia
ROP2 was overexpressed in the Columbia background, presumably.
Yeah.
Okay.
And you've got a wild type Feronia, right?
Mm-hmm.
So the control that it needs to be compared to is the wild type.
Okay.
So are the FAIR8 ROP2 overexpressors still dwarfs?
I'm not sure if you looked at that.
My guess is that ROP2 overexpression is, I mean, it's involved in so many things other than just ROS.
Yeah.
So.
But I think what they're trying to do is they're trying to ask, as the sentence on top says, does FAIR act through NADPH oxidase? So if you disrupt FAIR, you've failed to turn on ROP signaling, and you failed to turn on the oxidases. So they're at, that's why they're doing the comparison of FAIR8 versus the other one, because you're showing that ROP2 partially compensates for the loss of FAIR8. You lose the signaling to activate ROP2, but if you overexpress it, you partially recover that. So I think that the comparison is what Shanice was saying, the FAIR8 ROP2 versus ROP2. Maybe I'm wrong.
Well, I mean, I think the point is that if you overexpress the downstream effector, right, then you can reverse the phenotype.
You're basically bypassing the receptor.
The need for the receptor, yeah.
Okay.
But it is kind of interesting that they get the two lines, the ROP2 overexpression, the two lines are consistent for the pseudomonad population, but they're inconsistent for the ROS species. So maybe that's the problem with the quantification of ROS species.
I think it's also hard to overexpress ROP consistently because it's so tightly regulated. So unless they looked at protein levels and knew that the protein levels were similar between these overexpression lines, then I think it becomes really hard to quantify these things.
Okay. All right, so they then looked at correlation between, I don't have much time, so I'll kind of, they looked at correlation between ROS and Pseudomonas levels and saw that there was a negative correlation between both, right? So the more ROS you have, then the less CFUs the less bacterium is in the rhizosphere. All right, so I mentioned at the beginning that Ralph peptides is a ligand for FER. And this is just kind of the last experiment that they did, where they looked at Ralph 23, which negatively regulates FER function. And they looked at, I think, Ralph 33, which is closely related to Ralph 23. And they added 1 micromolar of this peptide, and they looked at Pseudomonas colonization. So here is the parental line, Ralph 23, they see a significant increase in CFUs. For the Ralph 33, they didn't see any difference as compared to their vehicle control. For FAIR 8, they saw, for the Ralph 23, they saw no difference. And the same is true for the Ralph 33, right? So I think this is just more evidence that, this is like really the reactive oxygen species that is inhibiting Pseudomonas in the rhizosphere. And then finally, they overexpressed Ralph 23 and then saw an increase in the CFUs as compared to wild type. Okay, all right, I'll wrap up. Sorry, I think I'm probably okay on time. All right, so they proposed this final model that I mentioned at the beginning, where peptides, these Ralph peptides produced from nematode pathogens or fungal pathogens, can inhibit FER, and then that can in turn negatively regulate ROP2, resulting in low levels of ROS inside the cell, and that can in turn result in the proliferation of Pseudomonas that we see in the soil. And that's more or less it.
So I'm confused. It's positive regulation of ROP2, right? You activate ROP2, which...
Yes, sorry.
Okay.
Yeah, I misspoke that. Yep, sorry.
Okay.
Yeah, and in this model, I mean, basically, these Ralphs are released by nematodes, pathogens of the plant, right?
Mm-hmm.
So the pathogen's trying to downregulate ROS production. But it has the added result that it enriches for Pseudomonads in the rhizosphere, right? And then the idea is then the next time the soil has more of these Pseudomonads, so it's a suppressive soil, and it helps keep other pathogens down.
I guess the thing
I found that, I mean, I understand the model, but I think they could have spent a little more time explaining it,
Yeah.
Why are Pseudomonads uniquely sensitive to ROS? Why are they the only bacterial species in the rhizosphere that respond to the reduction in ROS? I mean, I see that it's good for the plant because then they suppress pathogens, but why are they so much more sensitive, I wonder?
Yeah, and I mean, I just think it might just be a focus of the paper, but it certainly seems like other bacteria, right? So the result that they showed with Burkholderia, for example, being enriched in soil, is perhaps like another microbe that also might be sensitive to ROS. Yeah.
So I guess another question I have, based on what Mary Lou was saying, why is it, I guess I was a little bit confused between having more Pseudomonas in the soil for the next generation, how that would be protective. I guess I'm confused.
Well, I mean, beneficial Pseudomonads in the root, like the ones they've been looking at here, they actually induce systemic resistance, and then you get resistance up in the above ground parts of the plant against pathogens.
Oh.
And there's a whole other thing that they didn't talk about.
Oh, I see. So it's not even something that they're talking about here. Okay.
Yeah, well, that's one big effect is you get an ISR from these pseudomonads. But these pseudomonads also seem to be good at outcompeting some of the others.
Okay.
Yeah, it is not only outcome. Yeah, it is not only outcompete. They have an experiment where they added the pathogenic pseudomonas, like it can cause the disease with that concentration, and in the presence of this beneficial microbe, that pathogen cannot cause any disease. So somehow it is inhibiting the pathogenic effect of the pathogen bacteria. Maybe plant has
the fluorescent pseudomonads release these fluorescent compounds, right? The combined iron and withhold it from pathogens. There's a lot going on with these pseudomonads.
Mm-hmm.
So I think this diagram shows why quantifying ROS is so hard. So the ROS is generated into the apoplast, but when they're actually measuring the levels, are they measuring cytosolic ROS or are they measuring apoplastic ROS? Right? I think that's part of the complication of measuring ROS the way that they were measuring it.
Do you have a sense of the dye that they used is able to distinguish? I guess I'm not sure.
Yeah, I think the dye is a apoplastic dye. I don't know the long name of the dye.
I don't think it crosses the cellular membrane.
Yeah, it doesn't cross the cellular membrane. It is apoplastic dye, specific for apoplast.
So Shanice, do you know what project you're going to be working on yet?
I don't. Shenyang said he wants me to do some single cell transcriptomics on different microbes in the plant. So whatever that means, I need to defend my thesis first, then I can talk about that.
Oh dear. All right.
All right. Thanks, everyone.
Yeah, thanks, Shanice. That was great.
Thanks. That was
Thanks
great.
for picking the paper. It was cool.
Is this our last one?
No, I
One
think
more.
Swadee's
Unfortunately, it might be on hormones, I hear.
One
Yeah,
in this next
hormones.
week.
Oxen, your favorite, Mary Lou.
Oh, good.
Kill me
We
now.
did have a few slides with hormones on them. We were glad to see.
Yeah,
She did sneak hormones in, but we didn't dwell on it.
tiny bit.
Yeah.
All right.
All right. Bye,
All
guys.
right, see you next week.